Okay, thank you, Dave. So I'm just going to tell you a little bit of background about how the potato genome sequence was generated so that you can interpret it in the right context for your breeding programs. So the potato genome was sequenced by a consortium called the Potato Genome Sequencing Consortium. It was a group of scientists throughout the world that were interested in obtaining the sequence of potato. It just got published a month ago. It made the cover. This wasn't maybe the most flattering thing about potato, and that you have chips and fries, and that came out 3 weeks after the link between chips and fries and obesity was revealed. So anyway, so it's done and it's available, and I'm going to talk to you about how we've integrated this resource into the SolCAP project, which you're familiar with, and Dave will be talking a little bit more about. So I sort of have two hats. I worked on the Potato Genome Project, which was funded by NSF, and then I'm working on the USDA-funded SolCAP. So I'm sort of the bridge between the genome and application. So just a little story about how the genome was sequenced. Initially, it was decided to sequence this diploid heterozygous genotype. We'll just call it RH. It's a Dutch genotype. They had made the ultra-dense map with 10,000 markers between SH and RH. This was published some time ago. RH was considered to be the least heterozygous of the two parents, and a physical map was made by the Dutch group at Wageningen, and Sanger sequencing started. This project started in 2005, 2006, and this was before the advent of really efficient next-generation sequencing methods, and we started off with a back-by-back approach, and the estimation was that we would need to sequence 6,000 backs for full coverage of the potato genome. So the initial strategy was that we're using the RH back by back. It turns out, as we started to collect some of the initial data, we realized that the haplotypes in this diploid were quite divergent from each other. So the first thought was that we'd have two haplotypes. We'll just call them 0 and 1. We'd chop them up and make a back library, and then we'd sequence them and then assemble back the two haplotypes. That was our thought, but it turns out that that was actually very difficult to do because the two haplotypes differed quite a bit outside of the gene region. So the question then was, could we find a homozygous line? And this would reduce our assembly issues. There'd only be one haplotype that we were looking at, and essentially you'd have, if it was a double monoploid, you'd have your two haplotypes. You chop them up, and then it would be much easier to put them back together. Okay, so this line does exist. This was made by Richard Buell, who's here, and this is DM13516R44. So it's a double monoploid line that comes from group Ferreira. Clearly has reduced complexity for whole genome shotgun sequencing, and David Spooner had put this in Solanum tuberosum. So one issue about this clone is it grows very, very slow, and this is thought to be because of inbreeding. So in case you've never seen it, this is RH. These are tubers of RH, and this is DM right here. Okay. So we checked the flow, the size of the genome. It was 850 by flow cytometry, which matched what RH was as well. So we set out to have some goals, and I'm just telling you this to just give you an idea. Obviously, this is a draft assembly, but I wanted to make sure you understood that there are missing genes, there are some assembly issues, and this is important that you just don't go and obtain the genome sequence and think, it's 100% perfect. But our initial goals were that we wanted at least 95% of the genes captured in the assembly, including the regulatory regions, that we would assess this by looking to see how many of the Sanger express sequence tags that are already in the public database were present in the assembly, and we wanted 95% of these. And we wanted to have these anchored to the chromosomes. So we wanted at least half of the genome anchored, and that would have represented 80% of the genes. And we wanted to annotate the genome for genes. And we asked, I'll go through what a Contig size is, this N50. Essentially, we wanted the assemblies to be useful in terms of our interpreting gene structure. So we wanted an N50 Contig size of 15 kilobases, and so an N50 is where half of the bases would be found in a Contig of at least 15 kilobases. And we wanted a scaffold size, and I'll go through the relationship between a scaffold and a Contig. We wanted this to be much larger, and we wanted this to be at least a half a megabase. So the assembly process, I don't want you to worry about the precise mechanisms of this. What I want to focus on is that you take the reads and you put them into an assembly algorithm that then generates first contigs. And so these are smaller, kilobase size contigs, and then you add in some more sequence data, these paired ends and these mate pairs, and you build linkages between the contigs to form scaffolds. And then you add in even more information over here and fill in some gaps, and you're ending up with your final assembly, which is called a super scaffold, where you've linked the scaffolds together into a higher order of assemblies. So these are the three tiers. So this is the smallest, this gets bigger, and then this will be the biggest unit size in the assembly outside of anchoring it to a chromosome or linkage group. So we did do some quality control to see how good the assembly process worked. We took 10 of the DM back clones that represented about a megabase, and we compared the whole genome shotgun sequence, which is on the bottom, with the back sequence up here. And certainly there are gaps in the sequence, and there's some minor rearrangements here of misassemblies, but overall the sequence between the whole genome shotgun assembly and the backs were quite good. So there were no major gross misassemblies. We did another QC where we had sequenced two ends, the forward and the reverse end of a whole suite of phosmid and back libraries, and then we went back and asked independently how many of those are in the correct order and orientation and the correct distance. And so what's shown here is that if you take the forward and reverse end sequences from the backs and you ask how many of them are in the incorrect orientation, it's a very small percentage. How many have the incorrect direction? It's a small percentage. And you can look at that with the phosmids as well here. It's a very small amount of misassembly. And what's interesting, if you go ahead and look at the RH backs, because we had forward and reverse sequences for those, and you look at them on the assembly, it's a little bit larger, but this is probably because of rearrangements in RH versus the double monoploid. So the statistics of the assembly, this is called version 3 of the assembly. And the take-home points here is that we were targeting an N50 contig size of 15 kilobases, and we got a N50 contig size of 31 kilobases. So this is a pretty reasonable contig size. You know, the average gene is going to be less than 5 kilobases, so we're going to capture the full gene on the contigs. So the N50 scaffold size we were targeting was a half a megabase, and we ended up with a 1,300 megabase scaffolds, or 1.3 megabase scaffolds. And the total representation of the assembly is 727 megabases. So we're missing part of the genome. That's the most important thing for you to take home. Part of the genome is missing. Most of that's missing is the heterochromatin and the centromere. So we're missing a few genes, definitely, because there's gaps in the assembly, but the bulk of what's missing is the repetitive sequences in the potato genome. Okay? So one thing that's important for you, this group, is that there was no genetic or physical map for the double monoploid, but a subset of individuals in the PGSC formed a mapping group, and what they did was they started to anchor the scaffolds and super scaffolds onto a genetic map. And they used a whole series of markers, SNPs, SSRs, DARTs, and they had a population generated by SIP, a DM by DI population, and they started to anchor the scaffolds to the genetic map. So they also used a few other resources. They had a physical map from RH, they had some markers from the POMA-MO site, and they had some maps that were present in SGN. So pretty much they had experimental data they generated directly themselves, and then they used a suite of data that they collected from elsewhere. And they targeted that they wanted to get 90% of the assembly onto the genetic map. When they finished, they had anchored 86% of the assembly, and this contained 87% of the genes. So one thing that's important that you understand is that they've anchored these scaffolds, super scaffolds, onto the linkage map. But this doesn't necessarily mean that they're in the correct orientation. So they're definitely in the correct order, okay? But they're not necessarily in the correct orientation. So I wanted to go through a little cartoon of how this is. So if we think of a chromosome right here, and we've got a set of super scaffolds, one black and one red, they could be positioned in order on the pseudo molecules or on the genetic map like this. So this would be a head-to-head relationship. But if we only have one genetic marker anchoring this superscaffold or this superscaffold, it actually could be in a whole different set of orientations. So the orientation, if there's only one genetic marker placing this, it could now be head to tail, it could be tail to tail, or it could also be head to tail in this orientation. So that's what's pretty important. We don't necessarily have multiple markers on each superscaffold, we may only have one marker, and that places it in a position, but it doesn't orient that sequence. So it could be flipped around north to south on the pseudomolecule. So another activity inside the consortium that I was involved in was to generate a lot of transcriptome data, and I'm mentioning this because I think it's a really valuable resource for the community. So we annotated about 39,000 genes in the genome, and between my group and the Wageningen group, we sequenced 45 libraries of cDNA. 29 of these came from the double monoploid and 16 came from RH, and just to let you know, these are heavily focused on tuber development. So there's a lot of different stages of stolons and tuber development. The double monoploid libraries are more focused throughout the life stages and also on abiotic and biotic stress. So it's a nice collection of expression data for the genes in the potato genome. We did this all with next generation sequencing. We had over 740 million reads, and these were used for annotation of the genome to improve the structure of the genes, but mostly it's used for inference of the biological function. So one thing that we wanted to make sure people are aware of is that one question is how many reads did we collect, and was that enough to detect all of the expression that was going on in those tissues? So what's shown here are the different tissues that were sampled from the double monoploid. What's shown in red are how many RNA-seq reads we had in millions, and this is actually from the start of the project to the end of the project, so this is just a reflection of how much more yield you get on a Illumina sequencing lane throughout the course of the project. But what's shown in blue is how many genes we actually detected as expressed in the different tissues. The lowest one right here is, I believe that's tubers, maybe? Or no, that's sepals. And then this one up here is salt stress. So it's actually, there's only a very weak correlation between the number of reads you have and whether the gene's expressed. And once you reach a certain threshold of about 10 million reads, 5 or 10 million reads, you've sampled as many transcripts as you can. So for the DM that we did 29 different libraries. We had 600 million reads, and in this developmental time series, we detected expression of 60% of the genes. And we were able to make a hierarchical clustering of the different tissues and their expression patterns. What's shown over here are all the different fruit and flower libraries. Right here are roots and callus. Over here is stress conditions. And then over here is wounding conditions. And then here's the tubers and stolons. And then these are the biotic stress. So the other thing that we did is we compared RH to DM. We had not only the back sequences that we started with RH, but we also had some whole genome shotgun sequence for RH. And we compared the different haplotypes. And in one comparison where we compared 99 megabases of the RH back sequence to the double monoploid genome, we found that the overall sequence identity was 97.5%. There was one SNP every 40 base pairs and one insertion deletion every 394 base pairs. And if we looked within the RH, we actually found that the sequence identity was a little bit less, 96.5. There was one SNP per 29 base pairs and one indel every 253 base pairs. So for cultivated potato, now I'm going to tell you a little bit about the work that we did here on the SolCAP project. We sequenced the transcriptomes of 3 varieties using this RNA-seq method. And we got sequence from Snowden, Atlantic, and Premier Russet. And what we did here was we took tuber, leaf, flower, and callus tissue. We made normalized libraries, and we sequenced this on the Illumina flow cell, and we got 30 million reads for each one of these libraries. We anticipate here, we predict that we got 50x coverage of the transcriptome, which is really deep coverage for SNP discovery. So this was the purpose of this, was to do SNP discovery to design the Infinium chip. So we went through a computational pipeline. This is published in BMC Genomics this year. And what we did was we took our RNA-seq reads from those 3 cultivars. We also went to the public database and got the Sanger ESTs. And we went through some filtering to remove low confidence SNPs. We then aligned them to the genome to find a genomic position for the SNPs. And we ended up with a set of 69,000 SNPs that we're calling high confidence SNPs. So these are available. These have met the Infineum design standards, and these are available on the SolCAP website. I'll go through that in a minute. Okay. So the data is available for the genome, and you go to potatogenome.net, and that takes you to Potato Genomics, which we support that site. And essentially, you could download all the data. It's freely available. You can do a blast search, and then you can look at it in a genome browser. So what I want to show you is just a few screen capture shots of the genome browser, just to make sure you're all familiar with this. So you enter the site, and you can select a region. You can enter in a text here. You could search for something like a kinase or starch, and this would generate a return for you. You can also type in scaffold with its coordinates, and then this is the overview window right here. So this looks like it's a 4 megabase scaffold, and we're looking at this position right here. And then we see a gene overview region right here that's highlighted in yellow. And if we look at that in more detail, you can see here that there's actually 4 annotated genes. This one's an adenosine 3-phosphate 5-phosphate transporter. And then there's a gene here, this vacuolar iron family transporter, and a set domain containing protein, and then a Kelded motif domain protein over here. So these are the loci. These are the loci. And then we have different gene models to represent those loci. And so what's shown here is the BGI representative gene model. So this is the model that is the longest coding sequence. And then we actually have isoforms that we have predicted based on the transcript evidence. So you could see in this case over here, there's only 1 isoform. In this far case here on the left, there's 4 isoforms, and there's quite a few isoforms over here for this gene right here. We've also aligned the predicted protein of these loci to UniRef to get any similarity to proteins in the public database. And we've also aligned them to the Sanger EST. So if you wanted to look at a longer EST sequence, you could do that. And then what we've placed is 2 different tracks of the SolCAP Infineum data. We've placed our candidate Infineum SNPs. So these are the 69,000 very, very high confident SNPs that we detected from Snowden, Atlantic, and Premier. And then we also have only the SNPs that are on the SolCAP Infineum array. So here we have 5 SNPs that are high confidence SNPs, and only 1 of these is actually on the array, because the array only was able to hold a fraction of what we actually were able to predict. So we also have alignments of the predicted proteins to different dicot genomes, Arabidopsis, grape, and poplar. So what's shown here are called what's called the wiggle tracks, and what these are is the transcript evidence for that. So what these are, each one of these is a different library. So this is tubers, leaves, callus, wounding, and they're really just a heat map, intensity map of the expression that we detected using RNA-seq. So the darker the blue color means that there was more expression of that exon. So these are the exons of the gene, and if you stitch them together, you'll get the actual gene model. And so this is good to just do a quick look to see what tissues your gene is expressed in. It can also give you some information that maybe the gene model is not annotated properly. So other accesses, there's a blast search. It's supported with all the blast family. You could download everything. You could download the transcripts, and you could download the proteins. Okay? If you want to make your own genome browser, you can do that as well. We provide the GFF. So one thing that I think is important, the easiest way for us to provide the transcript levels to you is through an Excel spreadsheet, believe it or not. So this is actually in the Nature paper, and if you go onto the Nature site, you can just download supplemental table A and B. And what this is the expression levels. And this example here is from the RH tissue, and all this is a list of the genes that are expressed against each one of the libraries. And then here's the putative function. So you can download this for, this one's the DM, and then there's also RH as well. So you could download this, just have it on your desktop. If you're interested in a gene, you can pull this up and quickly look at its expression across all the different tissues. The only issue is these things are very large files, like 30 megabytes. It takes a long time for it to actually download. But I think these are very helpful for you to get an idea of expression. Now, the units for this RNA-seq data, they're normalized for the length of the transcript and also for the number of reads you got per lane. So it's in what's called FPKM, which are fragments per kilobase per exon model per million mapped reads. So just think of this as a normalized value that you can go down a library across many genes and also between libraries. So the SolCAP site also has some data that's accessible, and what we have is some intravarietal SNPs we detected in the Sanger population, but these aren't nearly as rich as our Infinium data, but we provide for download the 69,000 information as well as the 8,300 SNPs that are on the Infinium array. So you can just go to the SolCAP website, and if you click on this link right here, this is the 8,300 SNP mapping information and the context sequence. So essentially you get the SNP, which superscaffold it's on, what position, and then here's the sequence. And you can do the same thing for the 69,000 SNPs. So if you're interested in any candidate SNPs for your own work, you can just download that. That's freely available. So just some acknowledgments. The Consortium was a huge international team that were working together for the generating the potato genome sequence. I think the only other member here is Richard Fallu is the other member that's here at the meeting. And then on SolCAP, which Dave will be telling you a little bit more about, it's obviously a very large collaboration as well. So if there's any questions, I'll be happy to answer them. Yes, Richard.
So, the wiggle track.
Uh-huh.
You see a lot of variation in different exons for one gene model.
So just so everybody could hear the question is, if on the wiggle tracks for the different tissues you saw difference in intensity across the exons, most likely that means that there's alternative splicing and maybe that splice form is under or overrepresented compared to the other ones. So the first thing I think I would do is you look at the representative model versus the isoforms and take a look to see if an isoform is being predicted that has a skipped or a retained exon, and take a look at that, and then you could come back and make an interpretation. But I think there, if you're really specifically interested in it, you would need to remap the reads, and you'd have to do it a little bit differently because we mapped against just the representative isoform. Okay. Yeah?
When you compared the three genomes, you came up with one SNP every 30 or 40
Mm-hmm.
base pairs. Overall, if you look just at exons, is the number changed dramatically, or is it still quite high?
No, so in the coding region, it's much more conserved. The intergenic region is quite diverged. So yes, it's different. It's different depending on which part of the genome you're actually looking at. Yes.
Do you have an estimate for what that number might be?
No, but I think, let me look at this one slide. I actually think that these numbers here are for just the coding sequence. I think I'll have to go check that part. But the problem is in the intergenic region, you would just have missing sequence. That's the problem. They're so diverged that there would just be like 25 kb that was just totally missing, right, in the two haplotypes. So that's the issue. So I'm thinking that this would only be where they actually aligned well, which would typically only be the coding sequence. Because if they didn't align very well, you can't include them. And if you notice, if you look, see, so we actually had a lot more sequence for RH, but it didn't align very well, so you can't quite get those numbers. So I have to look, but I think these might just be coding sequences. I'll have to check. Maybe including the introns, but I'll have to check. Yes, Dave.
Go to the next slide. Well, keep going. It's the next one. Yeah, so there, you're filtering down from the over 500,000 SNPs through the RNA superreads, and also then you have the Sanger SNPs. You get down to 81,000 SNPs. So what was the filtering that was going on there to take it down to that extreme?
Okay, so for the remote people, this is the slide that's titled Generating High Confidence SNPs. And so after you do the filtering just on sequence quality and coverage, you're left with about 81,000 SNPs. But to make an Infinium SNP, it needs to be biallelic. You don't want that to be anywhere near the intron, right? And you also can't have too many SNPs in that 50 base pair window. So that's where we went from 80,000 down to 69,000. Okay? And that's just design criteria. So the 81,000 are actually high confidence SNPs as well, but they may have too many SNPs next to the one SNP you'd want to measure, and so you can't design an assay properly against those. Right. So here going from the 575,000, which are filtered. So there's several filtering that's going on. First, you start with over 2 million SNPs, and you filter first for read depth, density, and quality. But then what we had to do, this was from all 3 accessions, then we aligned them against the genome to make sure we weren't, miscalling things. So this is the total amount of SNPs, and then this is the unique number of SNPs. So there's plenty of SNPs, so you could do this for any accession. You just want to filter, filter, filter because you're going to have so many anyway. All right. Yes? Yeah, so the question was, is the orientation of the super scaffolds, and when is that going to be updated, and when would we know? So there is a new version of the pseudomolecules that will be coming out, I don't know, in a few months. But Dave will actually show you data that, at least in terms of it being on a linkage group, it's very, very, very correct. So that's not an issue. And I don't know when the new pseudomolecules come out that they can really, I don't know that they added a lot more markers, because that's the only way you can get the orientation, right, is if you have at least 2 markers and you actually detect a recombination event. But in terms of them being in the correct order, Dave will show you data from 2 diploid maps that they're highly accurate. I think there's only 200 SNPs that aren't on the right linkage group, right? So I think the pseudomolecules are actually quite good in that aspect. The orientation's only going to be resolved as soon as we get more markers to put on there, and so that Dave can tell you about. So we've added more markers now from the Infinium chip in terms of where they're at. Okay, but I think if you do have issues, you should at least maybe let me know and the people that are making the pseudomolecules, and we can correct it. But they've sort of poured over them, I think, in detail, using as much data as they have in hand right now. So, okay. All right. Thank you.